Morning students, today we will study megasporophyll in Cycas. Good morning students. Today we will study megasporophyll in Cycas. The megasporophyll of Cycas revoluta are large ranging from 6 to 8 inches in length. Each megasporophyll is divided into an upper broad leafy portion and a lower stalk-like portion. The broad leafy portion is a lobed rather pinnate tapering to a point. The megasporophyll therefore shows a marked resemblance to foliage leaf but are much reduced. The ovules are borne laterally on the lower stalk-like portion. They occupy the position of the lower pinnae on this part of the organ and vary in number from 2 to 10 per megasporophyll. The megasporophyll as well as the young ovules are densely covered with a soft brownish woolly hair, but these are lost from the ovule as they reach toward maturity, so that the ripe ones are orange red in color. The broad pinnate portion of the microsporophyll of Cycas racinalis is further reduced and shows only trace of pinnate structure. The number of ovule is also reduced. In still other species of Cycas, the megasporophyll has only a serrate margin and further reduction in number of ovule to a single pair. In fact, a series of type showing the progressive and gradual reduction in the foliar nature of the megasporophyll can be arranged among the member of Cycadales. The ovules are large, erect, orthotropous type and slightly flattened in the plant of the in the plane of the megasporophyll. Each ovule is enclosed in a thick coat, the integument. In Cycas, the integument is differentiated into three layers: the middle stony layer with a fleshy layer both inside and outside. The outer fleshy layer is of orange-red color and the stony layer is of light yellow. The inner fleshy layer is used up as the female gametophyte mature and is left as a dry membranous covering or layer united to the middle of the tissue, that is the nucleus, except near the apex. Owing to the presence of stony layer, it is hard to cut the ovule. The integument does not completely invest the ovule. The tissue within the integument is called the nucellus. It constitutes the body of the ovule. The apex of the nucellus grow up into a beak-like outgrowth, the nucellar beak, which project into the micropyle. Some of the cell of the nucellar beak disorganized to form flask-like hollow cavity. It is the pollen chamber. It plays an important role in the early stage of fertilization. The micropyle lead into the pollen chamber. The pollen grain germinate here within the nucellus, almost filling it is the enlarged megaspore or the embryo sac. It is filled with a tissue constituting the female prothallus or the so-called the endosperm, formed by the repeated division of the megaspore nucellus followed by free cell formation. Its cells are filled with starch grain. Toward the micropylar end of the female prothallus, just below the floor of the pollen chamber, are developed 3 to 6 archisporangia with 3 or as the commonest number. The egg or the oosphere, the neck of the archegonia, open in a shallow depression called the archegonial chamber, which is formed by the upward growth of the prothallal tissue lying above the archegonia. The archegonial chamber thus lie below the floor of the pollen chamber. It is filled with a fluid. The The nucellus of the ovule of gymnosperm corresponds to the megasporangium of the pteridophyte. The ovule differ from the latter is being enclosed in an integument which may correspond with the inducium of the pteridophyte. Thank you.